Hi everyone, welcome to the Royal Botanic Gardens seminar series. And before I start, I would like to acknowledge the Gadigal people of the Eora Nation, who are the traditional custodians of the land from which we're broadcasting the seminar, and pay our respects to their elders past, present, and future. So today it's my real pleasure to introduce Professor Andrew Barron from Macquarie University. So Andrew will share today about his work on the neurobiology and the behavior of bees and their brains. So Andrew did a PhD at Cambridge University and then did various postdocs at the University of Sydney, the University of Illinois in the US, and at ANU in Canberra, before starting a position in 2007 at Macquarie University. So currently, Andrew is a Australian Research Council Future Fellow and an associate professor. So thank you so much, Andrew, for agreeing to give this talk, and off to you.
Thanks, Hervé. And look, thank you for inviting me to present, and thanks for allowing me to present by Zoom. I'm actually up in Mount Victoria in the Blue Mountains at the moment. That's on Darug and Gundungala land. The reason I'm there is I'm looking after an 11-week-old puppy who is sleeping next to me right now. Will hopefully stay sleeping for the next half hour. But if you hear any snoring or if you hear any random biting, it's not me. And okay, now the other problem with the Zoom presentation is normally I'd like the presentation to be more interactive and take questions as I go through, but I can't see you while I'm in presenter mode. So if we can hold the questions till the end, I'll rattle through the content as fast as I can, and then we'll take the questions at the end. So I've been working all my life on this thing, which is the honeybee. And the honeybee is really unusual among insects because most people hate insects and have an innate phobic response to insects. But over the last 15 years, everybody has come to love the honeybee, which is ironic because if you're going to have a bad experience with an insect, a honeybee is one of the things you'll have a bad experience with because they sting. I mean, very few people have ever had a bad experience with a spider, but everybody hates them. Many people have had bad experience with bees, but everybody loves them. There's probably an entire PhD in that, but it would be quite a boring PhD. The other thing that is unusual about the honeybee is that everybody recognizes that honeybees are smart. And this has worked its way all the way through pop culture. So I used to go to conferences and people would ask me whether insects even have a brain. And it's not that long ago that people thought that insects were incapable of any learning whatsoever. But this is a list of all of the cognitive clever things that it's claimed that bees can do. And it's a really quite remarkable list. And what I've been realizing, this is why I'm glad to talk to the Botanic Gardens, is that this honeybee intelligence, this type of honeybee mind has been, I think, incredibly shaped by its lifestyle. This form of intelligence has evolved because of the challenges of the very specific lifestyle that a pollinating honeybee has. It's a central place forager, foraging on constantly changing ephemeral resources. And I think those challenges are what have shaped the bee's type of intelligence and driven the cognitive capacities that we see. So what I want to do in this talk is talk about the kinds of aspects of bee intelligence that I've been investigating and talk about how they're relevant to the bee as a pollinator and how they can help us understand how and why bees move pollen around the environment in the ways that they do. So this is what I'm obsessed with. This is the honeybee brain. And this is a really beautiful micro-CT image that we constructed of the honeybee brain. Micro-CT is basically extremely high-resolution soft tissue X-ray. And with that, you can very quickly scan a bee brain and then reconstruct it in 3D. It is a minute structure. This scale bar down here is 1 millimeter. So we have a millimetrically scaled brain. What I want to emphasize is it's small, but it's not simple. It's small, but it's complex. And it is massively modular and incredibly precisely modular. So we have these regions here. So these structures here. Can you see my pointer? Can someone just say yes if you can see my pointer? I might need to do something like that. These structures.
Yes, we can see it.
Great. Are dedicated to visual processing. And they specialize to receive and process input from the compound eye. This is actually the retina of the bee, and it's a part of the brain that attaches onto the compound eye of the bee. If the head were here, the compound eye would be right here and right here. Here, these two structures in pale turquoise, these are the antennal lobes. So they take input from the antennae. The antennae will be facing out towards us here, and these are dedicated for olfactory processing. And then the input from the senses feeds into these structures here. And we can see the cups of those structures, and I'll just highlight those. So these are, if we look at these structures at the back and take away the rest of the central brain, we're left with these structures called the mushroom body. And the mushroom bodies we know to be the regions of multisensory integration and learning and cognition in the bee. The multisensory inputs come into different regions of the input region, the calyx here, and they're called the mushroom bodies because they're mushroom shaped. The output is through the lobes here, of the stalks of the mushroom. And a region is where learning and cognition is taking place in the bee. And the reason that I love working on honeybees is this brain only has about 1 million neurons, and we haven't got a full connectome, but we know what most of them do. So that for something like this, like the mushroom body, we're able to abstract the structure of that into computational models. So our mushroom body is nothing more than a 3-layer network. There's sensory input. It expands out into what we call a high dimensional space. There's a lot more neurons within the mushroom body than there are feeding input to the mushroom body. There's an element of feedback with this PCT pathway, which collects the total activity of the mushroom body and feeds it back and feeds it forward. The output of the mushroom body is plastic, and the connections here are adapted by experience and adapted caused by neuromodulators released by reward and punishment. And the output collapses down to just a very few output neurons that control behaviors like approach or avoid. So then we have the output as a premotor layer. So we have a very, very simple 3-layer structure, and we can confidently say this is the structure of the mushroom body. And then we have a constrained model that we can interrogate in terms of how something like this can support the kinds of cognition that we're seeing in a bee. And what's interesting to me is this kind of 3-layer structure with an input layer, a very large middle layer in terms of neurons or nodes, compressing down to a very small output layer controlling just a few options. This is a classic structure in computer science and machine learning, and it goes way back. It used to be called a perceptron. It would now be called a 3-layer feed-forward neural network. This kind of structure is known to be great for solving classification tasks and for rapidly updating the behavior of the network based on the kinds of reinforcement that the network receives. So it very easily allows the network to classify things by outcome. Now that classification problem is really key for a honeybee. A honeybee has to make a living by foraging in this kind of environment. This is an Australian mixed meadow. Range of flowers available to a bee. The bee's survival and the colony's survival depends on the forager honeybee extracting pollen and nectar from these flowers at maximum efficiency to carry them back to the hive. To do that, the bee needs to be able to learn what each flower type offers and to be able then to adapt its behavior according to which flower type is offering the best reward and the rewards it most needs. And of course, no two flowers are exactly the same. So it needs to be able to classify flowers by meaningful types. And that mushroom body structure is exactly the kind of thing that we would need computationally if we were to solve a complex classification problem like this, where we're taking a huge diversity of purple type flowers, none of which are exactly the same, and within which there'll be subtle subtypes of flower age, and classify them according to the types that make a difference. Now, this isn't so much a scientific study, but it's a demonstration I did once about how good honeybees are at learning to classify by meaningful type. So I did a demonstration, I did it for the ABC actually, on the capacity of honeybees to learn artistic styles. So the challenge we set the bee was to distinguish the styles of 3 different landscape painters. We had Emily Carr, we had Monet, and we had Seurat. And what I wanted to ask was, how long would it take for the bees to distinguish paintings by artists? So they're all landscape painters. They're all using the same basic tones, and they're all painting things like trees and landscapes. but they all have very, very distinctive styles. And the question is, how long would it take a honeybee to learn to classify paintings by artistic style if we made the differences in artistic style relevant to the bee? So the way we did this is we trained the bees into an arena, and we first trained them to respond to tiny yellow squares. And we did that by loading the yellow squares with sucrose. Now, the bees' ecology means they are incredibly rewarded by drops of sucrose, and they are incredibly motivated to find small drops of sucrose in the environment. So this kind of pre-training is very, very fast and very easy for a bee. And we also stuck these number tags on a bee so that we can identify the individuals that are taking part in our learning test. Then once the bees have been trained like that, this is an example of one of the learning trials. We set an array of paintings inside the arena, 2 of each artist, and the bees saw one of the artists on entering the arena, and it had to hold that painting in memory and match that painting to a different painting by the same artist inside the arena. And to make it salient, we loaded the paintings that the bee had to, well, so for example, in this example, we put a painting by Monet outside the arena. And then on the Monet paintings inside the arena, we put a drop of sucrose. On the Emily Carr and the Seurat paintings, we put a drop of quinine, which is a bitter solution. So that made the difference between artists meaningful to the bee. And this is what a learning trial looked like. And oops, that's not what I want. Hopefully this will work. Yeah, here we go. So the bee sees Monet entering the arena, and this is training. So it's moving and sampling the paintings. It's visually inspecting them. On some it inspects and doesn't land. On some it inspects and does land. You can see how it inspects every painting. Here it lands on Emily Carr. That's bitter, so the bee immediately rejects and flies away. It inspects the rest of the paintings. It finds Monet, lands, and gets sugar, so it feeds. So that's the kind of way that we train this. So we're training that we show them a painting on entry, and it has to match the painting on entry to the same artist, different painting, but same artist inside the arena. Now we trained the bee for just 24 learning trials on this task with 3 different artists. This is how they behaved after training, so after 24 bouts of training. Here there's an Emily Carr painting outside the arena. In this learning test now, all of the paintings are loaded with sugar, but we want to see which painting the bee will land on first. And if it's learned the task on seeing Emily Carr outside the arena, it should choose a painting by Emily Carr inside the arena. Now watch after training, they inspect the painting on entry to the arena. They fly into the arena, and this is their behavior inside. They still visually inspect all the options available, but they're not landing until they find a painting by Emily Carr, they land and feed. So that's how the behavior changes with training. So that shows that they can learn to classify paintings by artist, but the real test was you want to give the bees paintings by the artist that they had not seen in training. So here we have novel pictures by Emily Carr, Monet, and Seurat that the bees had not seen in training. And in this task, we set a painting by the artist that the bee had never seen in training, and we see whether they can match that novel picture to the artist. So here they've got a new picture by Emily Carr, never trained on this picture. They inspect it. They actually land on it, so they're slightly confused by that. But they inspect it visually, enter the arena, they inspect the paintings inside the arena, and they land on the Emily Carr. So what that shows is the bee has learned to classify even novel paintings according to artistic type. So this classification capacity of bees is incredibly finely honed because it's an absolutely essential survival capacity for this bee to be able to classify flowers by natural type. But there's another problem to foraging on a mixed meadow like this. All of these different types of flowers will differ in the rewards they offer, the probability of the rewards they offer, and the nature of the rewards they offer. So this raises a different problem that the bee has to solve. Can it learn to specialize, to prefer the types of flowers that are most likely to give a nectar reward? Now, this problem with a mixed meadow like this, computationally, it's the same as what we call a five-arm bandit problem or a multi-arm bandit problem. You have multiple options to choose from. In the learning, you have no idea which of those options are rewarding or non-rewarding. So you have to sample across the different options while trying to learn simultaneously in those options which options is most likely to offer the reward and what the probabilities of reward are for each option. Now that's the task that we call a multi-arm bandit task. And to assess how well a bee can solve this kind of task, we set them what we called the Fivearm bandit problem. So again, we had an arena that a bee could choose to enter and fly in. And in the arena, we set out a patch of artificial flowers, which were just colored discs. And again, the colored discs were loaded with either sugar for reward or quinine for punishment. We trained the bee with 5 different colors. And we gave the colors a random ranking so that whatever color we decided was color 1, that was always rewarded. Color 5 was always punished. But for the other colors, whether they were rewarded or punished depended on what color they were paired with. So in training, we only ever gave the bees pairs of colors. For example, color 1 with color 5. And we rewarded the color with the higher rank, and we punished the color with the lower rank. So in this case, C1 was paired with C5, so we rewarded color 1, we punished color 5. If we had set up a task with C2 versus C4, we would have rewarded color 2 and punished color 4. If it was C3 with C5, we would have rewarded color 3 and punished color 5, and so on. And we gave bees every color combination apart from one that we saved for testing, which I'll mention in a moment. So what we were trying to do here, we were trying to ask, when bees have this kind of complex comparison, are they learning the rankings of things and learning the absolute reward likelihood? So are they basically comparing options and learning to rank them relative to each other because that information is available to them? Or are they simply learning not a ranking, but their experience of reward with each color? So are they not comparing, but simply learning the probability of reward of each color? This is what we wanted to ask. We trained the bees for just 18 trials in this very complex task. And that 18 trials let us go through every possible combination of colors twice. So they only saw any specific combination of colors twice. So it's really not that much training. And then we gave them a learning test, which compared the color that had always been rewarded in training with the color that was unrewarded in training. And this was simply to see had they learned anything at all. We gave them a transfer test, which is colors they had been trained on, but a novel combination that they had never seen before. So we're asking, can they transfer what they've learned about the rankings of these colors or their history of reward on these colors? And we gave them a novel test, which was one of the colors they had learned on with a color they had never seen before. Now they solved the learning test incredibly well. They learned this at almost 100%. So even just in 18 trials, they quickly learned to prefer C1 over C5. They also solved the transfer test better than chance, but their performance wasn't as great. They also solved the novel test. They actually preferred the color on which they had been trained over the novel color. If we looked at their learnings of the rankings, it looked like they learned the rankings pretty well. So here we've got the R index, which is the number of times they landed on the rewarded color, landed on the color when it was rewarded versus the number of times landed on the color when it's punished in training over total number of choices. And if we looked at their preference for the colors as it establishes over training, we can see they basically established the same rankings over training that they developed that we set for them. But this is the interesting thing. If we compared their performance in the transfer test, what we found is that their preference for the more rewarded color, which is C2, correlated perfectly with their experience of C2. So individual bees that in training had more rewarding experiences on C2, which are up here, had the strongest preference for C2 in the transfer test. And the correlation is extremely significant and explains most of the variation. So what that tells us is the bees are actually not learning to compare colors. What they're learning is their individual experience of each color, how many times it's rewarded, how many times it's punished. and then they're matching their behavior to their experience. So if they've learned that a color is rewarded 60% of the time, in the tests, they will land on that color 60% of the time. So what this tells us is bees are incredibly good at learning probabilities. And I stole a picture of your Mount Annan garden as a tribute to you, not because I'm plagiarizing your gardens, but because it beautifully illustrates a complex mix of flowers that a bee would forage on. Now, when you've got something as complex as this, ecologically, the most effective strategy is not to try to learn everything relative to everything else and to do a comparison relative to everything else. Simply learn your experiences on each individual thing and then match your behavior to your past experience. What that means is you will end up majoring on the flower that you have learned to be most rewarding, but it also means you don't exclusively use just one flower type. It means that you will also keep sampling other options in the environment. So this strategy of probability matching, which is what the bees show, and which they learn incredibly quickly, ends up being ecologically the most optimal solution when you're foraging on a complex, unknown patch of flowers. So bees are expert, rapid probability matchers. And what interests me is that they're learning this kind of probability task faster than vertebrates can learn it. And one thing I'm looking at now is how we can get this faster acquisition of this kind of complex task in a brain this small compared to a vertebrate brain. Now, the last thing I'll talk about is how bees move among flowers. And this will only take a couple of minutes. This is an experiment I did in a complex three-dimensional patch of borage flowers, because the movements of bees between flowers is critical for how effective they might be in transferring pollen between flowers. For this, we took a patch of borage, because borage flowers are nice because the bees gather pollen from them, and it's very easy to see them gathering pollen. And the borage flowers open and close and die off quite rapidly. So the patches, and it has this real three-dimensionality. And the patch is constantly changing. So we asked, are bees learning routes through the flowers, or is bee behavior in the flowers completely random? Now, the bee brain has this beautiful structure. This is not a honeybee, it's a megalopter bee. And I'm using this example because we actually do have a connectome for the specific region of the brain that processes space in the bee, and it's called the central complex. It's a minute part of the bee's brain, but it sits right at the heart of the bee brain. And I think that's for a reason. I think this is one of the oldest and most critical aspects of the insect brain. It has within it a structure here called the central body lower, which is an open ring, but it acts like a compass. It actually orients the bee's behavior relative to the world around it. The central body upper, which is this region here, helps to relate where things are in space relative to the bee. And this structure lets the bee organize its movements and behavior relative to things. What we did is we followed bees on multiple multiple natural trials through our borage patch. And we labeled every single flower in the borage patch, and we reconstructed that patch in 3D. And we were able then to track how bees localize their movements through the borage patch on each individual visit. Now, each of these points is not a visit to an individual flower. It's the center of a bee's movements in the borage patch for any particular visit sequence to be made in a particular visit. So each point is a different visit to the borage patch by a different bee. Each color is a different bee. What I want to emphasize is different bees specialized on very, very different parts of the borage patch, for the most part. Some partially overlapped. You can see that I've got to move that image there. B5 is partially overlapping with a subset of the region used by B3, but B3, B2, and B1 are extremely segregated. Now, we analyzed this, and I've got other days of data that shows the same kind of thing, different bees specializing in different parts of the patch. We analyze this in terms of, do bees have different routes? What's happening? Are they actively avoiding each other? None of that's true. What's happening is that the bees are entering the patch. Different individual bees have idiosyncratic points for entering the patch. What then drives their behavior is a nearest neighbor rule. From the point at which they enter, they start using flowers in terms of, I'll land on the nearest one, and then move to the nearest flower. The nearest neighbor rule dominates. So they're not learning idiosyncratic routes, even though it looks like they are. They're not avoiding each other, even though it looks like they are. What drives this behavior is that they arrive at the patch through very idiosyncratic directions and enter the patch at idiosyncratic points. And that then shapes their subsequent behavior. So the main points I want to make, just to quickly wrap up, is that bees have a brain that seems to have evolved specializations for incredibly rapid and effective classification, incredibly rapid and effective learning of probability, and a very, very refined spatial sense. And these three things together help the bees very effectively work flowers in extremely complex environments. And I think those abilities sit behind everything that a honeybee does in terms of its cognition. And I'll stop at that point. Thank you for your time. I'm going to stop sharing so I can see you, and I can jump back into the slides if we need me to.
Thanks so much, Andrew, for an excellent talk. Let me just stop the recording now.